We can date the age of the Earth in part by meteor fragments. And here we can see a chunk of a meteorite fragment that dates back to 4.6 billion years old. And we actually found this crater in Arizona, right here in the United States. We believe that life arose on Earth about 3.8 billion years ago. Changes in body structures and molecules have slowly accumulated through that time, producing the variety of organisms that we see today. Scientists use the geologic timescale to divide the history of the Earth into both eons and eras. And we can see on this figure from our book, all of these eons and eras are represented in this timeline. These periods are defined by major geological or biological events such as mass extinctions. Even though the events that led to today's diversity of life occurred in the past, many clues suggest that all organisms are in fact derived from a recent common ancestor. So we have lots of evidence for evolution on our planet, and this evidence tells us how species are related to one another. Both fossils and biogeographical studies provided some of the original evidence for scientists. Also, scientists could compare both embryonic development and similar anatomical structures, and that can also tell us how organisms are related to each other based upon similarities and/or differences. However, it was really when we figured out how DNA works that we had unprecedented clarity on how organisms are related. In the 1960s and 70s, scientists began analyzing sequences of DNA, proteins, and other biological molecules. Paleontology is the study of the fossilized remains or other clues to past life. As I mentioned previously, fossils provided the original evidence for evolution. Fossils form in many different ways on our planet, and depending on the way the fossil forms, it will preserve different types of organisms. For example, sometimes plant remains can fossilize because plant cells are surrounded by cell walls. More commonly, animals with skeletons, such as this fish or this dinosaur, are very easy to fossilize because of those nice solid bones. Even though fossil evidence is diverse, it is often challenging or even impossible to find transitional forms between groups as organisms evolve over millions or even billions of years. However, we do find some, and one example is this organism, Tiktaalik. Tiktaalik is believed to be a transitional form between aquatic amphibians and land-based reptiles. The fossil record is also incomplete, partly because many organisms are soft and their body parts simply don't fossilize. It would be very rare to find a fossil of a jellyfish or an earthworm because they don't have any bones or hard parts. Also, the Earth is constantly changing. Earthquakes and erosion and the movement of the tectonic plates in our Earth's crust might destroy fossils over thousands or millions of years. However, we do sometimes find fossilized soft matter. For example, we believe we have found a fossilized brain that's about 8,000 years old. If the fossils record the evolutionary history of our Earth, then we can use those fossils to tell a story based upon where we find those fossils. For example, high on a mountain in Oklahoma, in the middle of the United States, we find the fossils of organisms that only live in the ocean. Therefore, that tells us that Oklahoma, millions of years ago, used to be underneath an ocean, but over evolutionary time and due to geological uplift, those mountains were raised and those fossils now are high in the air, hundreds or thousands of miles away from any marine water sources. Dating fossils yields many clues about the timeline of our life's history. The simpler and less precise method of dating fossils is called relative dating, which assumes that lower rock layers have older fossils than the newer rock layers. Absolute dating uses chemistry to determine how long ago a fossil formed. One type of absolute dating is called radiometric dating, which uses radioactive isotopes. Throughout life, organisms accumulate both carbon-14, which is a radioactive isotope, as well as a more stable carbon-12. This carbon usually comes into our body in the foods that we eat, and that carbon ultimately came from carbon dioxide, but plants can take that carbon dioxide and build sugars. Living organisms have a constant amount of this carbon-14 embedded in their tissues. After the organism dies, however, they're no longer eating, they're no longer breathing, they're no longer doing respiration, so no more carbon-12 or carbon-14 gets added to their system. However, carbon-14 is radioactive, so it decays at a constant rate, leaving the organism in the form of nitrogen. Scientists have calculated that during any 5,730-year period, the amount of carbon-14 in the organism divides in half. In other words, the half-life of carbon-14 is exactly 5,730 years. By determining the amount of carbon-14 contained within a fossil, scientists can therefore estimate how long ago the organism lived. When you're studying for the exam, make certain you can distinguish between both relative and absolute dating. So we've talked a lot about fossils now. What about biogeographical studies? Well, biogeography is defined as the study of the distribution of species across our planet. The Earth's geography has changed drastically over the last 200 million years. In these pictures from our textbook, it shows us how the continents have changed and shifted over the last 200 million years. And notice that as drastic as these changes are in the landforms found on our planet, these images represent only about 5% of the Earth's history. Scientists hypothesize that this cycle has therefore occurred several different times. According to the theory of plate tectonics, the Earth's surface consists of several rigid layers, which we refer to as tectonic plates. And these plates move in response to forces acting deep within our planet. Both volcanoes and earthquakes are evidence for these tectonic plates. And in the diagram on your screen, it shows us a map of all of the plates, and you can see they push up right next to each other. And where they push up next to each other is where we often have lots of earthquakes and lots of volcanoes. Fossils help geographers piece together these Earth's continents into an original supercontinent, which we refer to as Pangaea. Here is a picture from another source that shows all of our current borders for our current countries on planet Earth, and here they are mapped onto Pangaea. It's just a fun visual representation for what it would look like 200 million years ago if we had our current countries back then. One of the famous biogeographical studies is Wallace's line. Animals on either side of Wallace's line in Indonesia have been separated for millions of years, evolving independently. This transitional zone therefore resulted in unique animal communities that are separated by a relatively small body of water. In fact, only about 22 miles of open ocean separates these hugely different geographical zones. When you're studying for the exam, make certain you can describe how the positions of the Earth's continents have changed over the past 200 million years. Investigators often look for anatomical features to determine the evolutionary relationship of two organisms. Two structures are said to be homologous if the similarities between them reflect recent common ancestry. All of these animals, the human, the bat, the bird, the lion, and the seal, all have similar bones in their forelimbs. These similarities suggest that their common ancestor must have shared the same bone configuration. It is important to note, homologous structures need not have the same function or look exactly alike in order to be considered homologous and in order to be considered a structure that reveals common descent. Instead, think about it this way. Different selective pressures in each of these animals' evolutionary lines have led to small changes from their ancestors' bone structure. Another structure that can reveal common descent is a vestigial structure. A vestigial structure has lost its function but is homologous to a functional structure in another species. A very common vestigial structure is the human tailbone. Another vestigial structure is vestigial hind limbs in snakes. Snakes no longer have 4 legs, but we believe that the common ancestor of snakes did have 4 legs, and evidence of this is that modern snakes still have vestigial hind limbs. Also whales. Whales have pelvic girdles, and pelvic girdles are usually only for animals that live life on land. It helps support their weight in the force of gravity. This tells us that the ancestor of whales lived on land, and over evolutionary time, whales adapted to live in the water. Anatomical structures are said to be analogous if they are superficially similar, but they did not derive from a common ancestor. For example, here we have a salamander and a small insect and a crayfish. All of these animals have no pigment, so they are essentially albino, and they have no eyes. These similarities arose by convergent evolution, which produces similar structures in organisms that do not share the same lineage, and they do not share a recent common ancestor. Instead, the selective pressures of living in a dark cave where there's no light has, over time, produced these traits. Lack of pigment, therefore, arose independently and by chance in each of these cave-dwelling animals. Presence of spines arose independently in these dry-loving plant species as well. These plants look the same, but on the tree of life, they are completely unrelated to each other. Here's a challenge question for you. The streamlined shapes of dolphins, which are mammals, and sharks, which are fish, evolved independently. Therefore, the body plan of these two animals are, your first choice is homologous, the next choice is vestigial, the next choice is analogous, the next choice is a product of convergent evolution, and the final choice is both analogous and a product of convergent evolution are correct. Please pause the lecture if you don't want to see the answer yet. And the answer is both analogous and a product of convergent evolution are correct. When you're studying for the exam, make certain you understand what homologies reveal about evolution. Anatomical similarities are often most obvious in embryos. Notice how much similar the human and chimp skulls are in fetuses compared to adults. In fact, as fetuses, the chimp and the human are very difficult to tell apart. Another example can be seen in adult fish, mice, and alligators. As adults, you would never mistake a fish from an alligator, but as embryos, they look very, very similar. This leads us to ask this question: how do similar embryos develop into such different organisms? One answer might be in homeotic genes. Homeotic genes control an organism's development. Small differences in gene expression might make the difference between a limbed and a limbless organism. The limbed organism that we're referring to here is a chicken, and the limbless organism is a python, which is a type of snake. Homeotic genes, therefore, help explain how just a few key mutations might produce a new species. When you're studying for the exam, make certain you can describe how the study of embryonic development reveals clues about shared evolutionary history. Comparing DNA and protein sequences determines evolutionary relationships in unprecedented detail. It is highly unlikely that two unrelated species would evolve precisely the same DNA and protein sequences simply by chance. Statistically speaking, it is much more likely that the similarities were inherited from a common ancestor and that differences arose by mutation after the species diverged from their ancestral type. In this example from our textbook, it talks about cytochrome C evolution. Cytochrome C is a mitochondrial protein that is often used in molecular comparisons. The more amino acid differences between species, the more distant the shared common ancestor. Notice that the chimpanzee has 0 cytochrome C differences from humans. Molecular clocks assign dates to evolutionary events. If a gene is estimated to mutate once every 25 million years, then we can assume 2 differences from an ancestor might arise in 50 years. Here we can see on our screen I'm highlighting the 25 million year mutation and then the 50 million year mutation. If we add these up, therefore, 2 species that derived from the same common ancestor just 50 million years ago might have 4 differences accumulated in the nucleotide sequence of this gene. And remember that genes give us our traits. Let's make sure we understand how to do these calculations. Your question is, mutations in a gene occur at a rate of 1 nucleotide every 10 million years. The gene sequence differs by 6 nucleotides between 2 related organisms. Therefore, how long ago did these organisms split from their common ancestor? Was it 2 million years ago? 30 million years ago? 60 million years ago? 120 million years ago? Or are none of the choices correct? Pause the lecture if you don't want to see the answer. The answer is 30 million years ago. When you're studying for the exam, make certain you understand how analysis of DNA and proteins support other evidence for evolution. As we've seen, evolution does not always lead to greater complexity. Sometimes features are lost. Snakes without limbs burrow more easily than those with limbs. Therefore, in snakes, natural selection favors the alleles that give them no limbs. Those snakes can hide better and survive longer. The case of the salamander that lives in a dark cave is slightly different from the snake. In an environment without light, pigment provides no selective advantage. Since producing pigment costs energy, alleles conferring colorlessness are favored in these cave-dwelling organisms. Our textbook chapter ends with an example of a snake species that lived 90 million years ago. We found the fossil of this species, and it has functional hind limbs, pelvis, and sacrum bones. Therefore, this is the most primitive snake discovered, and it supports our theory of the loss of limbs on land for snakes.